So today I have the distinguished pleasure of introducing to you Dr. Heather Proctor from Canada. So in our international series, last time we had Europe, this time we have Canada. And Dr. Proctor grew up in central Alberta, Canada. As an undergraduate in the Department of Zoology at the University of Alberta, she surveyed the diversity of water mites in the province. For her Master of Science at the University of Calgary and PhD at the University of Toronto, she continued her water mite theme and explored predatory behavior and sperm transfer behavior, respectively. During her first academic position at Queen's University in Ontario and second position at Griffin University in Queensland, Heather broadened her research to include bird mites, soil invertebrates, and ecological restoration, in addition to the water mites. In Australia, she and Dave Walter wrote the first edition of Mites, Ecology, Evolution, and Behavior. So then she returned to the University of Alberta in 2002. Even though water mites now make up a relatively small part of her program, she will still considers them her first love. So without further ado, welcome Heather, and we are very excited. We're fascinated by the video that you showed. So go ahead, Heather.
Hey, thank you very much. Thank you all for attending. I hope that this will be an interesting talk and that it won't be aimed too low. I'm aiming this at quite a general audience in addition to more hardcore water mite people. So when we first think about, how do people get interested in arthropods? Often it's very young, and often it's because we're interested in something that's bright and lively and colorful and big. And so butterflies are often the way that children get introduced to insects. Jumping spiders are often the way that people get introduced to the study of spiders, into araneology. And for water mites are the butterflies of the mite world. They are the gateway drugs to acarology. It's very easy to be interested in water mites because they are big, relatively, not that big, but big mites. You can see them with the naked eye, and they are really lively and brightly colored. So this has been noticed for a long time. Antonio Berlesi's famous alphabet included many water mites up to the letters that he managed to get through. In Soren Williamson's British Hydrocorrina, again, beautiful colored plates of water mites, focusing on their gorgeous and unusual bright colors. Another ray, not the Ray Society, but Ray Fisher, also has a beautiful plate of colorful torrenticulid mite dorsal plates, showing once more that the color is really exciting in this group of animals. And we can visualize water mites in different ways now with fancy techniques like colorized SEM. This is from Martin Oegerle's work. You might know him as Micronaut, and new ways that keep being discovered. This is Antonio Valdecasa's work showing that you can use a combination of UV light and extended depth of focus to do a really good mimicry of much more expensive laser scanning confocal microscopy to look at the morphology of water mites. So people for a very long time have been fascinated by the shapes and colors of these mites. But having said that, you may be asking, what are water mites? We know they're pretty, but what are they? I'm going to start by telling you a little bit about what they are not. So water mites are not the only aquatic mites. Mites have invaded freshwater and marine habitats many times, and there's representatives from most major taxa, Mesosigmata, Aurobatida, including Astigmatter, and then within the Prostigmata, we have two main groups that have invaded water, the Halichoroidea, mostly marine, and then finally the Hydrocnidia, the true water mites. Smith's recent Water Mites of the World indicates that there are almost 7,500 named species within this group now, and more keep being discovered every year. So Hydrocnidia are Prostigmata. But before we talk about the Prostigmata and then Hydrocnidia in particular, I want to give a tour of some other aquatic mites that are not water mites. in the strict sense. And I apologize to Tobias Pfingstl because I will not be looking at intertidal marine mites in this tour. I'll be focusing on freshwater mites that are found in the northern part of North America, which is where I am from. So that's where I have most of my pictures. So this is not exhaustive, but it will give a general overview. And again, I'm hoping that people who aren't mite biologists could be watching this. So some of this is quite elementary. So we can just skip over the things that you guys mostly know. So Mesosigmata, they are predators of small invertebrates and invertebrate eggs for those that happen to live in association with fresh water. They tend not to be subaquatic. They mostly walk around on the surface film. And in standing water in my area, there are some members of the family Bladysociidae that one quite commonly finds in standing water, Platyceus and Chiroceus, which often hitches rides on crane flies to get from pond to pond. Aurobatids are best known for being hugely diverse in soils and being very important components of terrestrial ecosystems. And here's a nice array of terrestrial Aurobatids. They are strongly sclerotized as adults. And one thing that you really notice when you flip them over is that Aurobatids have large anal openings. That's because they ingest particles, which is an unusual thing for chelicerates to do in general, most being fluid feeders. In the freshwater associated Aurobatids, none of them swim. They're mostly detritivores and herbivores. The most common genus in my area, and probably most places around the world, is the genus Hydrozetes. It is associated with duckweeds as a juvenile, or sometimes they feed inside individual duckweed plants. And they are spectacular for having this plastron of air that allows them to exchange gases underwater. There are other freshwater Aurobatids as well, though, including quite a diversity, even within Alberta, which is not exactly a biodiversity hotspot. And here you can see that this female appears to be ovoviviparous. She's got a couple of little babies developing in her. So Astigmatta are actually part of the Aurobatidae. They're derived within the Aurobatidae, but their ecology and morphology is quite different overall. So we often talk about them as if they were a separate group. They do share with the other Aurobatids particulate feeding with large anal openings. And overall, they look very much like juvenile Aurobatids. A very different thing, though, is that in the Astigmatta, the dudonymphs are modified for phoretic transport on larger arthropods. And this is a great thing if you're living in standing water as an aquatic astigmaton. You can get from place to place, even quite isolated places, if you happen to hop onto a flying insect. There's two families with common aquatic or semi-aquatic representatives in this area, or this area in my area. The Acaridae has several genera that are associated with freshwater, including this particular dudonymph here. You can see the sucker plate used for hanging on to a host insect, and the Histiostomatidae, which are interesting microbial feeders and have, you probably can't see that on your screen, tiny little serrations on their chelicerae. The Histiostomatids include a group of tropically associated aquatic mites that I don't have around here, the Nepenthes associated. So Nepenthes is this wonderful group of pitcher plants. There's a high diversity of Histiostomatids associated with pitcher plants, including a couple that are actually good swimmers. Well, probably the only other good swimmers other than the true water mites. So let's move on to the prostigmata on this tour before we get to water mites sensu stricto. Halocarids are a highly diverse group with almost 1,000 species, but most of those are marine. Only about 56 species of them are freshwater, and they are very tiny little guys, got a highly distinctive splay-legged stance like this. Like the Aurobatids, they are non-swimming. They could be predatory or algivorous. And we really know very little about the ecology of freshwater species. Most of what is known about halocarid biology is for marine taxa. Okay. Also within the prostigmata, we've got a few other oddball groups that are water associated. The Homocolligidae they look a little bit like army helmets, very, very round, heavily sclerotized and teeny tiny. They tend to walk around on the surface of water bodies, and we don't know really much about them at all. And then we've got the Sigmans. Most of them are not associated with water, but in northern North America, Eustigmaeus is associated with very wet moss or partially submerged moss, and they're actually moss feeders, which is unusual for Stigmaids to be plant feeders. Okay, now we have neared to where we are targeting water mites into the big clade Parasitengona. This is the clade that includes many superfamilies of familiar mites like velvet mites, chiggers, which you may have experienced if you happen to live in a warmer area than here. They're larval velvet mites, and Hydracnidia, the water mites in the strict sense. All members of the Parasitengona share a complex life cycle that includes alternating pupa-like stages. I'll show you that in an upcoming slide. And they also share the lifestyle of being parasitic as larvae and predatory in the post-larval stages. So very complex life cycle. In addition to water mites in the strict sense, there are other aquatic parasitengones, including many members of the Johnstonianidae. They are actually moderately well understood, the ecology of at least just Johnstoniana rapax. It's quite unusual in that the larva can act either as a predator or a parasite. An interestingly shaped member of the Parasitengona that's also associated with soggy moss is the Calyptostomatidae. They can completely retract their gnathosoma, which gives them this funny blunt-nosed appearance. The larvae actually look similar to adults. This is unusual for the Parasitengona, and they are particularly common as parasites of tepulids. Dudonymphs and adults are predators of larval tepulids. And you don't really need to know that Calyptostoma occurs in Alberta, but I was very pleased to know that myself. So the other group before we get to the water mites in the strict sense is this very mysterious family, the Stygothrombidiidae. They're skinny, wormy, red or orange soft-bodied mites that live in interstitial gravels in very often very fast, cold running water. The larvae are parasitic on stoneflies, and the post-larval stages are presumably predators, but to my knowledge, no one has ever observed them preying. I could be wrong. I'd be pleased to find out if I am wrong. They differ from true water mites in that they've got a median impodium on their claw, and they also have trichobothrial setae, whereas true water mites do not have them anymore. So Stygothrombidiids and water mites in the strict sense, which are all these guys here, may be sister taxa. So just popping out that part of the cladogram there. Here's the Stygothrombidiidae as sister to everybody else, but you can see that the support is not super strong. I would hope for more genes and more taxa to be studied before we can pronounce Stygothrombidiids or anything as sister taxon to the Hydracnidia. All right, finally, we're here in the water mites in the strict sense, in the Hydracnidae or Hydracnidia, or sometimes Hydracarina. The common name is water mite. They are the dominant mites in freshwater with a few marine species as well, around 7,500 named species in the world. And North America has around 2,000 of these. They've never been included in a single compendium though. So if you're looking for the big boys water mite book of North America, no one's ever written it. And indeed, half of these species probably haven't been fully described either. They occupy all types of freshwater habitats. And in my experience, they're most diverse in permanent lakes that themselves have a diverse array of macrophytes, of vegetation, and in permanent streams that have diverse substrates, cobbles, gravels, and fast and slow flowing water areas. So morphologically, water mites are highly diverse. They range from completely sclerotized little crunchy armored tanks to very soft and baggy and able to change their body shape easily, but they are never furry. If you find a red furry mite in the water, you have found a terrestrial velvet mite that has fallen in. Water mites are not furry. They can get up to 7 millimeters long, bigger than a fruit fly, but most are between 0.5 and 1 millimeter in length. They come in a gorgeous array of colors, red being the most common, but also green, blue, yellow, purple, any color of the rainbow you can think of, actually sort of almost shown in these 3 mites up here. Water mites can be those colors. And later on in this talk, I'm going to focus again on the colors of water mites and what adaptive purpose they may have. Most water mites have spectacularly well-developed eyes for mites, sometimes mounted on either side of an eye plate. And their legs may or may not have swimming hairs, which are long setae that basically tell you if you have swimming hairs, that's a swimming mite. If you don't have swimming hairs, you're a crawling water mite. Water mites have a very distinct swimming style, and I hope that people can see this video of a whole bunch of things swimming in the sample that I got from near my house. And there you can see a water mite, very distinctive, the way it flails its legs around, and it's easy to pick out from the movement of the other taxa. Life history of water mites is the classic life history of the Parasitengana. We start out with eggs that hatch into larvae that search for a host. The larvae will then parasitize, typically aquatic insects. They may search for the hosts above the water. They may search for the hosts below the water. And if, sorry, Ray, you're going, what kind of life cycle? What species is this? It is not a species. It is a whole bunch of species thrown together just to be a generic life cycle. Okay, yep, I'm sorry. I thought I should point that out. And then after being on the host and engorging, they will drop off and fall back into the water and engage in what's called the calyptostatic protonymphal stage. Calyptostase is a pupal-like, or rather a puparium-like stage in which an individual molts, but stays inside the cast skin and then molts again. So it's covered essentially by 2 cast skins, and then the active individual bursts out. So the protonymph is inside the skin of the larva. Then the active predatory deutonymph develops inside the skin of the protonymph, breaks out, is predatory. We've got another calyptostatic stage, and then we've got the predatory and sexual adults. Some species skip the parasitic stage and go straight here, and that's a very interesting area of study, is the selective forces behind giving up parasitism, the advantages and disadvantages. As far as how those eggs get made, Hydrocnidia are all sexual and have separate sexes. There's no hermaphroditic water mites, and they have a ridiculous diversity of sperm transfer methods: direct via copulation, like these guys here, Mite-y Eulerian, indirect via courtship, but spermatophores are placed on a substrate and the female picks up the sperm herself. I'll show an example of that coming up. Or no courtship, males just put down spermatophores and leave, and females may or may not pick them up. There is, in this video here, around this part, you can see a bunch of Limnesia males depositing spermatophores. I won't show that right now, but maybe if there's time, I'll show it later. Here is some more information on sperm transfer in water mites if you're interested. So once the eggs have been fertilized, they're laid in discrete clutches that range from 1 egg to thousands of eggs, covered by a jelly coat. You can see clutches of eggs here in this particular image, and the jelly coat eggs are glued to a substrate. There are a couple of exceptions though: Hydracna oviposits in vegetation and Uninicula oviposits in sponges or mollusks, and the eggs in these taxa are laid individually and are not in a coat. One interesting thing in water mites that has not really been investigated, and I would sure be interested in doing that sometime myself when I have time, is why is communal oviposition so common? Very often you'll see a whole bunch of females ovipositing in exactly the same area. Why? Is it like the best piece of water plant to oviposit on? Or is it just that, she started it and everybody else thinks safety in numbers, we'll add our eggs there too. These communal egg masses can be so abundant that they can be seen from the sky, as happened in this case. David Whartonby, who's a biologist in Alaska, spotted this big red slick on the top of a pond as he was flying over in a float plane. And when they landed, they saw that the slick was not an oil slick or some other horrible pollutant, but it was probably millions or billions of Ilias larvae. Just imagine how many would be in that original oviposited mass. Why? I don't know, but it's spectacularly interesting to investigate. So once the larva has dropped off the host, we've got the active post-larval stages, dudenymphs and adults both being predatory, and they very often feed on the same thing. Dudenymphs and adults tend to feed on the same thing. They can eat, depending on the taxon, insect larvae like the Limnocharies eating a Carotenoid, insect eggs, insect pupae, small crustaceans like Daphnia, and frequently other water mites. Water mites can be very fierce if you've ever observed them hunting and feeding. They often attack animals much larger than themselves. And they're able to kill and eat them because of their digestive enzymes. They bite, inject, subdue the prey that way, and then drink the liquefied tissues. And this particular author of a book about the powers of the creator displayed in the creation was really impressed by water mites. I'm just going to read this because it's an excellent quote. He said, "Hydracnae are among the most rapacious of living animals, bold, fierce, and cruel, the natural and inveterate enemies of all their congenera. They are no less hostile to each other, against which is waged a permanent war of extermination. Neither do they hesitate in attacking such animals as are suitable to their appetites, though double the size of their assailant." So even back then, people were amazed by the predatory behaviors of water mites. And because of this rapacious and fierce behavior, they have been considered for biocontrol of mosquitoes, both through predatory behavior of the adults and dudenymphs and parasitism of larvae on mosquitoes. But sadly, water mites are difficult to mass rear as biocontrol agents because you have to co-rear their hosts along with them, and it's difficult to keep the life cycles equally, well scheduled. So I was fascinated by predation, and that's what I worked on for my MSc, the predatory behaviors of water mites, particularly predatory behavior of the Unionicollidae. I really liked Unionicola crucipes, so this is me many years ago in a shirt that I made with Unionicola crucipes on it. And that's another member of the Unionicollidae. They are unusual in water mites in that they are sit and wait predators quite often. They will just stand around waiting for prey to come by them, and they recognize the prey moving past them by vibrations in the water column. So here's a Unionicola going after a Daphnia, and here's a Neumania that has caught a copepod. So when I went to do my PhD, quite to my surprise, it turned out that predatory behavior and sensitivities to prey vibrations turned out to be important in the mating behavior of some water mites. So this was one thing that I worked on, the mating behavior of Neumania papillator. In this group, or in this species, the male basically wiggles his leg at a female at the same frequencies as a copepod. And she will respond to that if she's hungry by grabbing the male, trying to bite him, but he is the wrong shape for her to be able to bite. And so he releases her. Once he's been grabbed by a female, he says, aha, that's a female, not just something that smells like a female. And then he deposits a group of these elaborate group of spermatophores, fans his legs over top of them, presumably sending a current of pheromone-laden water towards the female. And the female, if she's interested, will drop down, rub her venter over top of these spermatophores. They're sticky, they get stuck, the sperm packets get stuck to her ventral side, and then she brushes them into her genital opening herself. So that is a type of sperm transfer that is indirect, but because the female does this, the actual transfer of the sperm, but involves very close courtship and pairing of the mites. Also, while I was watching the mites, these mites sit around waiting for prey to come by, whenever I adjusted the light of the microscope, I saw a really weird thing happen. The eyes of the mites moved back and forth like that. Oops, too fast. Back and forth. Oh, my animation was too fast. Let's try that again. Back, forth, back, forth. And why? And also how? How do water mites move their eyes in a wiggly, jiggly fashion? It reminded me of the jiggly-eyed jumping spiders that are popular on YouTube and Reddit. Here is a video of one. Jiggly-eyed jumping spiders apparently move their eyes by actually moving their retinas, not the eyes per se, but it looks like the eyes are jiggling. In the water mites that move their eyes, they really actually are moving their eyes. How can they do that? And also why? So when I was doing my master's, I was trying to figure out how important vision was for water mites to be able to, capture their prey. And by experiments, the answer was not very. But I also did some sectioning of water mice to look at the structure of their eyes, and scanning electron microscopy. And to my great surprise, for some of the water mites in the scanning electron microscopy, I could no longer see the eye capsules, the lenses. I thought, where did they go? Did they fall off? They should be in the outside of the head. But not always. In some water mites, the eye capsules are indeed on the outside of the cuticle where sensible arthropods put their eyes. But in others, the eye capsules are sunk deep inside the gnathosoma of the mite, so that there is no external evidence of the presence of the cuticular lens. And this was discussed in 1905 by Lang in German, which luckily I could read. And so I learned all about water mite eye morphology. This is a cross section that Lang did of the eye of a hydrodroma, and it is on the outside of the head, or the gnathosoma. And here we have a chitinous part that goes inside the tissues, and there are muscles that attach to it that can allow the eye to be moved slightly. But this example down here of a Piona, the eye is completely sunken in to the gnathosoma of the animal, but it still has muscular attachments. So in my scanning electron, or in my histology, I also found similar things. That's a hydracna nymph. This is a drawing I made showing the attachments to the eye lenses. And here, this is a Unionicola showing their lenses are not on the outside of the cuticle at all. So there's numerous mysteries associated with this. And if anybody is of an anatomical bent, go for it and find out why. Why are some eyes sunk inside the heads, per se, of the water mites? And why do they jiggle their eyes? What's the point of that? I don't know, but it's fascinating to think about. Back to the ecology, moving backwards actually to the ecology of the larvae. As larvae, water mites can parasitize all orders of aquatic insects, except, to my knowledge, again, happy to be corrected, except for mayflies and the megaloptera, like Dobson flies, possibly because mayflies have that additional winged molt, which would be problematic if you are out of the water and your host molts on you, and megaloptera have terrestrial pupation. But other things have terrestrial pupation and still manage to get water mites on them. So that's not a completely satisfactory answer. Chironomids, non-biting midges, are particularly popular as hosts. And here I am showing a Coccolithidia mosquito that is carrying probably Aurornis water mites, but they weren't identified. There are some exceptions to being parasitic on aquatic insects. One is Larviothermacharis may feed on amphibians. There's not a lot of observations of this, so that's something to check out next time you're at a hot spring. And also, although most adults and deutonymous are predatory, there are some exceptions, especially within the Unionicolidae. There are quite a few of them that feed on tissues of mussels and snails. Here's a Unionicola species mite that is on the gill of a viviparous snail. And there has recently been found, mainly through a lot of work by Tom Goldschmidt and his colleagues, is that there are some Hygrobates species that parasitize salamanders. So here's a Hong Kong warty newt. And here, if you pop open the mouth of this guy, you see these little things embedded in the tissues of the newt's mouth. And these are Hygrobates water mites, post-larval stages of them, presumably feeding. So what about the opposite end? Who eats water mites? They are sometimes eaten by insects, like this water scorpion, Renatra, that had caught and sucked out the contents of a hydracna nymph. And soft-bodied water mites, as we saw from that quote before, they're very frequently eaten by other water mites. And sometimes you see soft-bodied water mites that have scars on their body, presumably from previous attacks from other water mites. But water mites are rarely eaten by fish, and red species appear to be particularly distasteful to them. And this brings me to a research vignette, a rather old research vignette, but I hope you'll find it entertaining if you haven't read it previously. So a little bit of background about color before we get into the research vignette on water mites. Bright color in animals and plants can have many different functions, sexual display, attracting pollinators or frugivores towards you, warning rivals that you're dangerous, or as in this compilation, recent compilation, warning predators that you are dangerous. This is, of course, called aposematism, and it involves signaling to a potential predator that you are distasteful, heavily armored and not worth the effort, venomous, poisonous, whatever, some reason why that predator should not approach you. Eulerian mimicry is a subset of aposematic evolution, in which there's independent convergence of the same sort of visual signal in different taxa. And the idea is that if there is a consistent aposematic signal from many different taxa that co-occur, then that's an even stronger enforcing behavior enforcing thing for local predators. If they learn that this yellow jacket wasp is dangerous, then anything that looks yellow and black stripes, like, oh, don't want to go near you. So the cinnabar moths are distasteful. The yellow jacket wasp is dangerous with its sting, and that's an example of Eulerian mimicry. When we look in freshwater, though, we don't see bright colors very often. Bright color in freshwater animals is rare relative to marine ones. Most freshwater animals look like this: brown, 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 yellow, green, brown. Pretty dull, except, as we know, for water mites. Water mites are a striking exception. Bright reds and orange are very widely distributed, but as I mentioned, there's other colors as well. And the bright red and orange ones really stand out clearly against the background green vegetation. So if you are brightly colored and your predators can see you very well, presumably if they've got color vision, maybe redness and orangeness is an aposematic signal in water mites saying, we taste bad. So that's not a novel thought. A couple of famous ecologists have thought that before. Elton in 1923 argued that bright colors in water mites evolved through Eulerian mimicry. In other words, he thought there was convergence in different lineages of water mites towards the same signal indicating distastefulness. And Kerfoot also thought that water mites advertised their distastefulness through color. He found that if he gave fish live red mites, they would spit them out. He found that if you dried up some red water mites and made them into powder with flour, the red flour balls were more distasteful to fish than powder made from yellow colored mites, supporting the idea that redness advertises distastefulness. And in a very small survey, he found that where there were fish present, in 6 ponds with fish present, the red mites were the more abundant than where the fish were absent, suggesting that red mites were able to survive in fish present ponds better than non-red mites. So from my personal observations, I had a different opinion from Kerfoot's statement. Based on my observations, I thought that red water mites were not more common in fish present water bodies. And also, I thought that Elton's suggestion of Eulerian mimicry implies, as I mentioned, independent evolutions of redness under the pressure from fish predation. But many terrestrial relatives of water mites are also red, like this velvet mite here. So redness, maybe that's the ancestral state, not independently evolved repeatedly in water mites. If redness is ancestral, I hypothesize, maybe distastefulness is too, in which case we should be able to take terrestrial velvet mites, put them into flour balls, try to feed them to fish, and fish should spit them out, indicating that the redness and distastefulness may be preceding the evolution of aquatic lifestyles in this group. So that was what my very, very first master's student, Nira Garga, did. This is Nira now, not Nira then. I set her to test Kerfoot's ideas with a much greater sample size of fish present and fish absent water bodies. So she had a really good sample size of those. And then she took the live mites and sorted them into color groups within genera using paint swatches, actually. So we just matched them into, like, evening mauve or coffee time, all those sorts of things, to figure out what was the dominant color of each mite. And after doing that and some stats, we found exactly the opposite of what Kerfoot had observed. We found that red water mites dominated the fish absent water bodies, with an average of 89.7% of mites in fish absent water bodies being red compared to in fish present water bodies, where only about half were. So yes, that's the issue. Not what Kerfoot observed at all once we bump up those sample sizes. So redness is associated with the absence of fish, not with the presence of fish. And even for genera with a range of colors, we found that the red or orange species were more common in the fish absent water bodies. So in the fish absent water bodies here, for these 4 genera that have variation in color, we found the majority of them, except for Acarology, were red when the fish were absent and not red when the fish were present, even strongly so for the Acarology. So that is not what Kerfoot saw at all, and doesn't support a role for fish predation as something that maintains redness. What about distastefulness? We ordered Dinothrombium magnificum from Arizona, there it is there, as our example of, can we test whether fish find the potential ancestral state of water mites to be distasteful. We used this bluegill fish as a predator, did the same sort of methodology as Kerfoot, and then tried to replicate Kerfoot's study with also drying up and grinding the distasteful red mite, Limnocharies. And we found that terrestrial velvet mite powder was horribly distasteful to fish. Even at something as low as 16%, almost 100% of the flower balls were rejected for the terrestrial mite powder. In fact, they were even more distasteful as ground up powder than were the Limnocharies, which were the most distasteful water mites in Kerfoot's study. So that was also interesting. Mites or fish do not like this food item, and they would never have been able to have a selective pressure on them. However, when we fed live Limnocharies to the bluegills, they did not like them. They spit them out. So drying them up did reduce the efficacy of the taste. So let's have a new hypothesis. We have no evidence that fish predation is the current selective force for bright coloration in water mites, and that redness is actually more common where there aren't fish. We find that terrestrial relatives of water mites are distasteful to fish, so that redness and distastefulness probably ancestral. And that suggests that redness and distastefulness preceded the invasion of water by the ancestral water mite, and water, instead of selecting for redness, maybe the aquatic habitat allowed diversification into different colors. We never see blue terrestrial velvet mites. We only see red and orange ones. It's in the water that we see this diversification. So that's interesting. But why red and distasteful at all in water mites and in velvet mites? So most scenarios of the evolution of aposematism say you start with the defenses. I taste bad. And then it's like, oh, I need to advertise this distastefulness with bright colors. So this sort of pattern. But there is another possibility. Bright coloration can evolve first for a reason other than signaling, and then distastefulness evolves second because you are so obvious. What is the original function of bright color? Well, for redness in particular, literature on pigments in Parasitengona revealed that many carotenoids in water mites and terrestrial parasitengons, it's very widespread. Red and orange pigmented Parasitengona have a lot of carotenoids. What can carotenoids do other than say, I am red? Many studies of planktonic crustaceans show that carotenoids are photoprotectants. They prevent light-induced oxidation of sensitive molecules. And pigmented crustaceans are more common in water bodies that experience intense solar radiation, such as alpine water bodies. And studies have shown that copepods with high levels of carotenoids can survive a lot more radiant energy than pale colored copepods. But the trade-off is they get seen and eaten more rapidly by fish. So let's move back to the mites now. Is there any evidence that red water mites occur in water bodies where they're more likely to have intense solar radiation? My personal observations are that such red-dominated water bodies are often very shallow, but they're also often very shaded, like vernal water bodies in the woods. So not very consistent support there. But when we think about the ecology of water mites and other parasitogons, the larval stage has very different exposure to light, potentially, than the post-larval stages, because the larvae are associated with the outer bodies of arthropods, and their hosts themselves may be exposed to a lot of light, both while on the host insect and while searching for the host. Especially true for those water mite taxa that run around on the surface of the water to search for hosts, rather than those that search for hosts underwater, because of course UV is attenuated inside the water. So here is a scenario, which we're getting close to the end here. I'll try to go pretty fast. Selection for carotenoid protection was in the terrestrial ancestors, carotenoids to protect from exposure to the sun, either during the larval stage or during the post-larval stages, as well as the larval stage. Bright coloration is conspicuous, and that distastefulness evolved as protection against visual predators. And then when the ancestral parasitogone that became the first water mite invaded water, the invasion of the aquatic habitat released some lineages from the need for carotenoid protection, because water itself screens from carotenoids. But not all of them. Would be some water mite lineages that would still need carotenoid protection, perhaps because their larvae were on a very exposed host. And mites with more UV exposed larvae can collect carotenoids through their diet to pass on to the offspring via eggs, even though the adults themselves may not need UV protection. Their offspring do, and so they collect the pigments and then give them to their offspring. So an undergraduate student, Michael Podruski, did an honors project testing some of these ideas. He wanted to find out whether red mites are actually red water mites, more tolerant of UV exposure than non-red mites. And so he used this particular fluorescent bulb to test this idea. It puts out some UV light, but it also puts out some visible light, and that could be a problem later on. So we used a Limnesia species. We thought it was one species because there was no morphological difference other than color, yellow versus red. And he exposed them to UV light and then counted, saw how long it took for them to die. And with the expectations that the red ones would take longer to die than the yellow ones, and the difference would under the UV light. So we did find that overall, both yellow and red did die faster under the UV light treatment, and that the red ones survived slightly longer than the yellow ones under the UV treatment, but there was not significantly different. Not a great sample size though. Michael also wanted to find out whether the color of the mother of water mite was similar to the color of the eggs. In other words, were pigments passed on to offspring. And he looked at mites from species in 7 genera, and he found that when the female was red, the eggs were always red. For the non-red water mites, in general, non-red females like this blue and white Neumania here tended to lay non-red eggs. Not always, sometimes a white water mite will lay red eggs. Here you can see, sorry, this is an example of a non-red laying non-red eggs. There she is, there's her eggs, very white. But sometimes ones that were non-red could still lay red eggs somehow. And how would that happen? If you're not red, how do you make red eggs? The assumption that red mites have carotenoids and non-red ones don't is not necessarily true, because keratinoproteins often appear green or blue, but they are still carotenoids and can potentially be turned back into redness. So the last thing that Michael did was look at water mites of different colors and examine their pigments with HPLC to look for carotenoids. And he found that every water mite, regardless of their apparent color, had a carotenoid of some sort. So yes, non-red mites could actually have good carotenoid protection, potentially from UV regulation or UV light, but we can't really tell that for sure until we do a few more behavioral tests and to find out whether they can survive just as long as red ones under UV. So that has never been published there. You're the first to know about that. But I hope that, having said it, I will encourage some of you to go and study the function of colors and whether carotenoids and different colors of pigments help to protect water mites against ultraviolet radiation. So we need to find out more things. Using the full spectrum of natural UV, we used a very partial spectrum of UV. Need to focus on eggs and larvae because that's the hypothesis that the colors are to protect them. And we really didn't study survivorship of eggs or larvae under UV light. Could try and find out whether we can manipulate the color or carotenoid content of postlarval mites through diet. That Limnesia undulata of yellow and red form suggests that maybe their diet could have caused the color differences, but we don't know. It could also be that that's a genetic difference. And finally, and good advice for every scientist in the world, keep an open mind and acknowledge the potential for multiple purposes of pigmentation. Not everything is necessarily going to be just for protection from UV, just for defending yourself against predators. And for all we know, maybe they sometimes use their color for courtship. I don't know. So on that note of not knowing, I will open the floor to questions if there are any. Thank you very much for your attention. I will stop sharing.
Okay, Heather, thank you so much for a wonderful presentation. Patricia, do you want to say something too? You're muted.
No, I forgot to introduce myself. Heather, no, this was fantastic. For those in the audience, I'm the president of the Acarological Society just for this year, follow the big shoes of Lorena. And I was supposed to say that before, but I have to leave. But I put a question in the chat, so maybe Lorena can tell me later the answer. All right, yep. Bye everyone. Thank you. Bye.
Okay, let's start with the questions. So we had a lot of interest on your presentation, which is great. And we had some questions beforehand. So one of them is, if you could clarify a little bit about the common methodologies for collecting water mites.
That's an excellent question, since I didn't talk about that at all. In general, you need a finer meshed dip net than is usually used. Most people, if they are general freshwater invertebrate collectors, they'll use a 500 micron mesh net. For water mites, you should probably aim for something like 250 microns, because many water mites would just go through a 500 micron mesh net. So you need a fine meshed net. And then if you are collecting in standing water, the best way to collect is to, I wish I had a net here, I don't, but pretend I have a net. And you would take your net and use the net itself to disturb any aquatic vegetation. And you make one sweep past it, turn the net around, and then go through the area of water that you've disturbed, because you'll sort of have knocked water mites off the plants that they'll be sitting on. They sort of perch on plants quite a lot, like birds, swimming occasionally, but not all the time. If you're collecting in running water and it's a rocky substrate, then you collect in the similar way that you would for any stream collection, which is you stand in the stream, you walk backwards, you shuffle the substrate with your feet, and then you continue to walk backwards upstream, and the water will take your disturbed substrate into the net. You can also collect water mites by taking aquatic macrophytes, and especially mosses. If there's submerged mosses, that's a great thing to collect. Take the aquatic macrophytes, let some of the water drain off so that it's not really, really wet, and then you put them into Tulgren funnels. So you can get some really interesting water mites and other strange things by doing Tulgren extractions of macrophytes. I found several genera of water mites in Alberta that I had never found before, simply by doing moss extractions. So that's a very good way of doing them.
Thank you. Another question. So we have a question. It's all unionicolous species oviposit in sponges or mollusks?
To my knowledge, yes, they all oviposit in the tissues of sponges or mollusks. The ones that oviposit in sponges are not parasitic on the sponges in their later life stages. Those that oviposit inside mollusks, most of them that are known are from the Unionoidea, which is a superfamily of bivalves, so freshwater clams. Some of them will just use the mollusk as a place to lay eggs. And the larvae and the post-larval stages are all, out in the water column, and the females just come back to lay eggs. But others of them, the larvae are parasitic on usually chironomids, but the post-larval stages, the deutonymphs and the adults, are parasitic on usually gill tissue inside the mollusk. So quite an amount of variation.
Okay. Next question. Is studying the biological aspects of life table parameters of water mites groups hard, or are there like basic requirements?
It is harder than for many terrestrial groups, like studying, say, phytosaids or stored product pest Astigmatta or spider mites, simply because of the need to co-maintain a culture of the host insect. And for that, you have to make sure that the life cycles are synchronized, because the larvae are only going to get on a particular life stage of the host. And then you have to, somehow allow I'll just say it's really hard to do with the water mites that are parasitic as larvae. The best understanding that we have about things like sex ratio of offspring and number of eggs produced come from studies where the water mite is one of those that has given up parasitism as a larva, and then you can rear them in the laboratory just with the post-larval food source. Like for the Aranyurus that have given up parasitism, Bruce Smith has reared cultures of them for decades on ostracods. And has studied a lot about their biology that way. So it's sort of sad, because it's the atypical water mites that are the ones where you can study their population dynamics in the best detail.
Got it. Next question. Dog. So have you collected in caverns any water mites?
No, I have not. Alberta is a very non-cave-like place. I think we only have like one cave system. So there's not a lot of opportunity for me to do that around here. But I expect that if people were to collect in like the limestone karst type environments, there should be quite a few water mite taxa there, provided that there was some way for them to get in there originally. So probably in caverns where there is an aquatic outlet that connects with springs or rivers that are outside the cave. That would be a way for evolutionarily those water mites to have got in there. But yet it's relatively, to my knowledge, understudied, but I may just be missing all of that literature, since I tend not to read the cave literature.
Just to clarify, are colors of mites used in their taxonomy?
No, not for water mites, at least not anymore. It may have been used early on, but certainly there's growing evidence now that you can get multiple color versions with the same CO1 barcode gene region. And in that, the barcode says this is one species, but you're seeing multiple variants of color in it. So we really are unclear about how heritable the color patterns, well, not color patterns per se, but the ground color of water mites is, and whether it's a good feature. But there are some generalities we can say, that there is, some families of mites are pretty much almost always red, orange, or yellow without any other variation, like Hydrodroma, for example, is you never find blue ones. So in that case, yes, that part's heritable, but when we get into the fancier colors, it becomes a little less clear.
Hey, can freshwater mites be found in vernal pools? And if so, what happens when they dry up?
Ian Smith has a great book written on that topic of what happens in temporary pools. Yes, there are water mites that can survive the drying up of water bodies, and it's typical that the one of the stages will be able to survive in the substrate while it is dry, and they'll overwinter as well in that state. I can't remember off the top of my head whether it's more commonly the deutonymph or the adults, but it's one of those two.
Thank you everyone for all the questions. We still have questions, so just
I can hang
wanted to
around.
say that. Okay, so how water mites respirate, communicate? And is there a report regarding that?
How do they respire?
Respired, thank you,
Okay.
and communicate.
Okay, so we'll talk about gas exchange. Water mites are quite interesting. For the larger bodied ones, you can often see that they do have like a tracheal system running through their body. You can see little tubes that appear to be gas filled, but they don't have openings to the outside. So they don't have open spiracles. They presumably use the just direct movement of oxygen in through their cuticle and then carbon dioxide out. And they very frequently help this exchange of gases by fanning over their dorsal side. So if you were there at the beginning of just sitting in the waiting room or at the beginning of the talk, you may have seen the video running in the background. Most of those mites were just madly fanning water over top of their back, and that would freshen the oxygen in the water and help to move it along. How do they communicate? Well, some communicate by vibrations, like that Neumania, which was waving its leg at the female, and they presumably mostly communicate via pheromones. There's good evidence of especially spermatophore-associated pheromones in water mites that allow females to detect where spermatophores have been deposited. Whether they communicate by stridulation or other sort of interesting methods, I don't know. I don't think that's been looked at, but it's not out of the question.
Right, I think I missed one in the chat. So this one's about what are my eyes? I think Ray already helped with part of the response with the answer. So have they, the eyes, been internalized multiple times?
Unfortunately, we don't have enough cross sections through water mite heads to know what the distribution is. It seems that possibly just once, maybe in the ancestor of the Hygrobatoidea, that they've been completely internalized, but I would be hesitant to go out on a limb with that. I hope someone will study that. That's a whole PhD's worth of investigation there.
So much to work on water mites. Okay, much diversity of water mites. Which diagnostic characteristic is mainly used for identification or important to keep in mind?
Okay, so identification of, is it a water mite? That's a hard one, but identification, differentiation among water mites, among the most important things are the morphology of the palp and the arrangement of the cocci and the structure and of the genital area and the number and arrangement of genital acetabula. So you do a lot of looking at genitalia when you do water mite taxonomy. Very species level diagnostic, genus level diagnostic. I would, in the talk, once it gets posted, you can go back in the talk, and there's a couple of citations for Proctor et al. 2015, and that is a book chapter in a book called Thorpe and Kovich's Freshwater Invertebrates, and that has a very big chapter on how to collect and how to identify water mites. So if anybody is interested in getting that chapter itself, you can just email me and I can send it to you.
And just as a reminder, this recording is going to be, this presentation is going to be posted at the Acarological Society of America website next week, so you can rewatch it. Okay, another question. I'm curious about how colorlessness adds to the color evolution story. For instance, terrestrial cave dwellers that evolved from normal red velvet mites are often colorless, and deep digging water mites are often colorless. If color is just for taste, then one would suspect it will be just as useful in cave dwellers, for example.
Yeah, so actually it was seeing some very white colored velvet mite larvae on nocturnal crickets that made me think, oh, maybe the redness is associated with exposure to the sun. And yes, so that's true. In cave dwelling, terrestrial Parasitengina and in the interstitial water mites, very often they're pale or colorless. But it's difficult to tease apart. Is that because they don't have to worry about ultraviolet radiation because they don't go out in the sunlight, so they don't need to worry about being damaged by it? Or is it because we don't need to protect ourselves from predators because they can't see us because it's in the dark? So if you can't see colors in the dark, there's no real point in being colored. And the third possibility is if the prey you're feeding on in the dark themselves don't have carotenoids because they're eating things. Carotenoids can only be made by plants. So for a water mite to get carotenoids, it has to eat something that itself, at least at one point, ate a plant. And so for cave dwelling organisms, the base of the food web may not be plants, it may be bacterial, in which case there may not be any bright carotenoids to pass along. So this is where one needs to do a lot of manipulative experiments to try and find out what's going on. Just looking at the patterns, unfortunately, is not going to allow us to differentiate amongst the different hypotheses very well. So you're muted.
Thank you. Okay, this will be the last question since we already passed 4. So about the evolution of parasitism in Parasitengona, how we explain the parasitism of that species on salamanders?
That's really, I would turn you over to Tom Goldschmidt on that. He's written quite a few papers on, he and his colleagues have written quite a few papers on them, and I confess that I have not read them all. So I'm not entirely sure why that lineage, it's a number of species within the genus Hygrobatids that are salamander associated. So I don't know what would have led to that at all. But again, an interesting question. And I'm just going to put my email address here for anyone who wants to ask me for more literature. Hopefully these chat things don't show up because I don't want everybody in the YouTube world emailing me. This is just for you guys here. There.
Yes, that's fine. That it's just the presentation that it's going to be posted. Okay. So that's perfect. Okay. Thank you again, Heather, for a wonderful presentation. Thank you everyone for attending. Stay tuned for next month for our next speaker. And again, next week, this will be posted in as a website. Okay. Thanks again.
Thank you. Bye, everybody.